Welcome back to this next video, and in this video we are going to talk about the neuronal signaling or the synaptic signaling, and I'll be focusing on how the neurons work. In my previous video I told you about the general structure of the neuron and the important parts of the neurons. In this particular video I want to focus on how the neurons they work. I hope the explanation in this video it does not get too complicated, but it is important to understand how neurons do what they do. Now there are many details, but you should go slow and look at the figures to get the concept of how the neurons work. Now, much of what we know about how the neurons work that come from experiment on the giant axon of the squid. Now, this giant axon extends from head to the tail of the squid, and that is used by the squid to move its tail. Now, how giant is this axon? It can be up to 1.5 millimeter in diameter, and what this 1.5 millimeter in diameter means that you can easily see it with your naked eye. Now, a lot of things they are needed to understand the whole concept of the neuronal signaling, and how the neurons work. But I'll not go through all of those things in this particular video. In this particular video, I'll be focusing on one of the most important concept to understand the function of neuron, that is the action potential. And in the next video, then I'll be telling you how this action potential is actually responsible for the release of the neurotransmitter at the axon terminals, and hence the transfer of message from the presynaptic neuron to the postsynaptic neuron. And in the action potential, I'll be focusing on the resting membrane potential, the depolarization, the repolarization, hyperpolarization, and threshold. And then in the end, I'll be slightly touching about the sodium-potassium pump, which are very important in the functions of the neuron. Now, when you talk about the neurons, they send messages electrochemically. What is meant by this electrochemically term? Now, this means that chemicals, they cause an electrical signal. Now, chemicals in the body are electrically charged when they have an electrical charge. And when you talk about these electrical charge substances or chemicals in the body, they are called as ions. And the most important ions, when you talk about the nervous system or the function of the neuron, they are the sodium and potassium. Both the sodium and potassium have got one positive charge. Another important ions in the neuronal signaling, they are the calcium. They are very important in the release of the neurotransmitter at the axon terminal, which I'll focus in the next video. And the chloride ions, which have got one negative charge. This calcium have got two positive charges. This chloride have got one negative charge. And there are also many some negatively charged protein molecules. Now, the combination of these positive and the negatively charged molecules or chemicals in the body, that is actually responsible for the functions of the neuron. Now, it is also important to remember that the nerve cells, just like the other cells in the body, they are surrounded by a membrane that allows some ions to pass through and block the passage of the other ions. Now, this type of the membrane is all known. It is known as the semi-permeable membrane. It is going to allow only those ions into and out of the cell which are required for the functions of the neuron. Now, one of the important things to understand is when you talk about the function of the neuron is the resting membrane potential. Now, when a neuron is not sending a signal, that means it is not involved in transferring of message from the presynaptic to the postsynaptic neuron, we say that a neuron is at rest. Now, when a neuron is at rest, the inside of the neuron is negative relative to the outside. If this is the inside of the neuron, and if this is outside of the neuron, so the interior of the neuron, that will be negative charge, the exterior of the neuron, that will be positively charged. Now, although the concentration of different ions, the positive and the negative ions, they attempt to balance out on both sides of the membrane, they cannot, because the cell membrane, because of its selective permeability, allows only some ions to pass through the channels, which are known as the ion channels, and I will be focusing on these ion channels at the end of this video. Now at rest, the potassium ion that can actually cross the membrane easily, as you can see over here, the potassium can passively move in and out of the neuron, but the chloride and the sodium ions, they have a more difficult time crossing. Now the negatively charged protein molecules, which are represented by A over here, they are inside the neuron and they cannot cross the membrane. Now, in addition to these selective ion channels, which allow the potassium ion to freely move, but not the sodium and the chloride ions and the proteins, there is a pump that uses energy to move 3 sodium ions out of the neuron for every 2 potassium ions it puts into the neuron, and this is known as the sodium potassium ATPase pump, which I'll be talking about at the end of this video. Now finally, when all of these forces balance out, the function of the sodium potassium pump, the selective permeability of the cell membrane, when all of these forces they balance out, the difference in the voltage between the inside and outside of the neuron, when that is measured, you have the resting potential, and this resting potential is also known as the resting membrane potential. And the resting membrane potential of a neuron is about minus 70 millivolt. As I've told you, that the interior of the neuron that is negatively charged, the exterior of the neuron that is positively charged. So when you talk about this resting membrane potential, it is actually minus 70 millivolt. What this minus 70 millivolt means is that the inside of the neuron is 70 millivolt less than the outside. Another important thing to keep in mind is that when the neuron is at rest, there are relatively more sodium ions outside of the neuron and more potassium ions inside that neuron. If this is a neuron at rest, and if you use these electrodes to measure the potential of the membrane, you will see that the in the resting membrane potential, that will be minus 70 millivolt, or the interior of the neuron, that will be minus 70 millivolt. So what is action potential? Now the resting potential, that tells us about what happens when a neuron is at rest. When you talk about this action potential occurs when a neuron sends information down an axon away from the cell body. In my previous video, I've told you that the axons, they are actually the extensions of the cell body, and they are responsible for taking the message away from the cell body towards the axon terminal. So the action potential, it occurs when the neuron sends information down an axon towards the away from the cell body towards the axon terminal for the release of the neurotransmitter, so that the message can be transferred to the postsynaptic neuron. Now this neuroscientists, they use other words such as the spike and impulse for the action potential as well. So if you look at the literature and you find these terms spikes or impulse in place of the action potential, they are referring to one and the same thing. Now what happens in the action potential is that there is an explosion of electrical activity that is created by a depolarizing current. Don't worry about depolarizing current, I'll come to it in a while. Now this means that some event when there is a stimulus that causes the resting potential to move towards the 0 milliwatt, or to move towards the positive value. Now when the depolarization reaches about minus 55 millivolts, a neuron will fire an action potential, and this minus 55 millivolt value is called is the threshold value. Just like as we say that minus 70 millivolt is the value for the resting membrane potential, this minus 55 millivolt is the value for the threshold. Now what happens is, if you look at over here, if this is the resting membrane potential at minus 70 millivolt, if there is a stimulus reaches at the neuron, what happens is that if this signal is strong enough, if the stimulus is strong enough to pass this threshold value, which is the minus 55 millivolt, that means that the action potential that will be generated and the resting membrane potential that will move from minus 70 millivolt up to plus 30 or plus 40 millivolt. Now, if the neuron do not reach this critical threshold, if a stimulus is there, but that stimulus is not enough to cross this threshold level, there will be no action potential. Also, when the threshold level is reached an action potential of a fixed size that will always fire for a given neuron. Now the size of the action potential is always the same. It doesn't mean that if this is a neuron, for example, that sometime for the action potential to fire you will need the value of -55, sometime you will need the value of -40 milliwatt. That is not the case. The size of the action potential, that is fixed. If the stimulus is strong enough to pass this -55 millivolt value, there will be action potential. So there is no concept of the big or small action potential in our nerve cell. All action potential are of the same size. Therefore, the neuron either do not reach the threshold or a full action potential is fired. This is known as the all or none principle. What this all or none principle mean that whether there will be an action potential or there will not be an action potential. Now this is what is happening in the action potential. If you look at over here, this is actually the nodes of Ranvier. This is one node of Ranvier, this is another node of Ranvier, this is another node of Ranvier, and between the node of the Ranvier, as I've told you in the last video, this portion of the axon that will be myelinated. That means that will be covered by the myelin sheet. Another important thing is that the ion channels, they are only present in these nodes of Ranvier, and there are no ion channels in this myelinated area of the axon. Now at rest, as I've told you, there are more sodium ions outside the neuron and more potassium ion inside the neuron. What happens is that when the stimulus that crosses the threshold value, in that particular case there is opening of the sodium ion channels which are present at these nodes of Ranvier. Now when these sodium ion channel opens, this means that is the sodium ion concentration is high outside the neuron, they are low inside the neuron, so the sodium ion, they will move into the neuron because of the high concentration outside the neuron and low concentration inside the neuron. Now these sodium ion channels, they are actually known as the voltage gated sodium channels. What I mean by this voltage gated sodium channel is that the opening of these sodium ion channel that are because of the voltage and not because of the binding of any ligand. Therefore they are known as the voltage gated sodium channels. Now during this change, when this sodium ion channel is open, the polarity of the membrane that actually move to a value of plus 40 millivolts. Now remember, as the sodium ion, they have got a positive charge, so when these positively charged sodium ions, when they will move inside the neuron, the inside of the neuron that will become positive, and when this happens, we say that the phenomena of the depolarization has occurred. So the movement of the interior of the neuron from the negative toward the positive value, that is known as the phenomena of the depolarization. Now what happens is that when these sodium ion channels they open in the interior of the neuron that become positive, it takes longer for the potassium channels to open. When the potassium and potassium channels they open, the potassium rushes out of the cell because the potassium ion concentration is high inside the neuron, that is low outside the neuron. So when the potassium ion, as they have got positive charge, when they will move from the inside to the outside of the neuron, this means that the inside of the neuron again that will move towards a negative value because you are removing the positively charged potassium ions from the interior of the neuron. Now this phenomena, when the interior of the neuron that move from the positive towards the negative value, it is known as the repolarization. And this repolarization that is achieved because of the opening of the potassium ion channels and the closing of the sodium ion channels. Now what actually happens is that when you are removing these potassium channels from the interior of the neuron to go towards for the repolarization, the action potential actually goes past minus 70 millivolt, and when the action potential that pass the minus 70 millivolt, for example, that move towards minus 80 millivolt, this phenomena is called is the hyperpolarization. And this hyperpolarization is achieved because the potassium channels they stay open a bit too long. in the end, what happens is when the phenomena of the depolarization has happened, the phenomena of the repolarization and the hyperpolarization has happened, the resting membrane potential of the neuron, that will be again achieved by what are known as the sodium-potassium pumps, and I'll come to that in a while. If you look at over here, there is positive charge outside, there is negative charge inside of the neuron. Because of these you can say these changes in the movement of the neuron, you can actually see that this positive one that will become negative, the interior will become positive, but very quickly that is again achieved. The interior is positive, the exterior is positive, and the interior will be positive again. Now, how the sodium and the potassium redistributions occur, because as I've told you that at the resting membrane potential, the concentration of the sodium is high outside the neuron and it is low inside the neuron, as well as the concentration of the potassium is high inside the neuron and that is low outside the neuron. So once an action potential has passed from a neuron, and when you come towards the resting potential, how this redistribution is achieved again, so that the sodium concentration is again high outside and low inside the neuron. Similarly, the potassium concentration is high inside and low outside the neuron. Now, this phenomena is achieved by the sodium-potassium ATPase pump. Now, if you talk about the name of this particular pump, this is actually dealing with the sodium, potassium, and this particular pump is ATPase in nature. What I mean by this ATPase, I'll tell you that in a while. But the sodium-potassium ATPase pump is actually responsible for the redistribution of the potassium and the sodium ions until the membrane potential is back to its resting potential around minus 70 millivolt. And why this minus 70 millivolt is necessary to get back again? Because it is at this point that the neuron is once again ready to transmit another action potential. Now, what happens is, as I've told you, when the sodium ion channels open at the start of the action potential during the depolarization, the sodium ions, they move inside the neuron. Now, what happens is during this redistribution, the sodium-potassium pump, it binds 3 sodium ions, and at the same time it binds 1 ATP molecule. And using the energy of the ATP molecule, it is actually removing these 3 sodium ions from the interior to the exterior of the neuron. As it is utilizing the energy of the ATP, therefore we call this pump is the ATPase pump. During the transfer, it is actually going for the hydrolysis of the ATP into the ADP, and when that one phosphate group that is removed, that will actually be responsible for moving the sodium ions outside the neuron. Now, the splitting of this ATP, it's provide energy to change the shape of this particular channel. As you can compare the shape of the shape over here, when there are no sodium ion attached and no ATP is bound to that. But when the sodium ions, they attach to these sodium potassium pump and the ATP that has been degraded, the shape changes, and this shape change is actually responsible for moving the sodium ions outside the neuron. Now, when the sodium ions, they are released to the outside of the neuron, so the new shape of this particular channel actually binds the 2 potassium ions which are outside the neuron, and when they bind over here, these potassium, they are actually taken back into the neuron, thereby re-achieving the resting membrane potential, which is the minus 70 millivolt, and that one phosphate group that will be released into the interior of the cell. So this is how the sodium potassium pump that redistributes the potassium and the sodium ion channels. Why it will move towards the minus value? Because you are removing 3 sodium ions, 3 positive charges, and you are taking back only 2 positive charges. So when this process continue, you will be getting a negative value, that is minus 70 millivolt, which is actually the resting membrane potential. This is an overview of what we discussed. when there is stimulus, and if that stimulus is strong enough to pass this threshold value, there will be the phenomena of the depolarization, and the sodium ion that will get into the neuron, thereby the interior of the neuron that will be positive, like plus 30 or plus 40. When that happens, then the phenomena of the repolarization start, in which you start moving the potassium ions out of the neuron for this process of the repolarization. But as the potassium ion pumps, they stay open for a long time, so you pass this minus 70 value and you go for the hyperpolarization, in which the interior of the neuron that actually moves toward minus 80 millivolt. But by the action of the sodium potassium ATPase pump, again you get this resting membrane potential of minus 70 millivolt, so that the neuron can again be used for another action potential. Now, one important concept about the action potential is what is known as the saltatory conduction. As I've told you in the structure of the neuron video, that when you talk about the axon, most of the area of the axon that is covered by the myelin sheath. So we call that is the myelinated area of the axon. The action potential that only occurs in these gaps between the myelinated sheath, and these gaps, they are known as the nodes of Ranvier. And in these node of Ranvier, there are the ion channels which are then responsible for the action potential that we just discussed. There is no action potential in this myelinated area of the axon because there are no ion channels. So what this saltatory conduction then means? This means that once the action potential that is being created in this part of the axon, in this node of Ranvier, that is going to jump from this node of Ranvier directly into the other nodes of the Ranvier, and then it will jump over here, then it will jump over here. That means that the action potential is actually spiking or jumping from one node of Ranvier to another node of Ranvier, and this movement is actually called is the saltatory conduction. As you can see over here, when the message is received and the message it travel down the axon, so there will be ion exchange in this node of Ranvier, then it is going to jump to this node of Ranvier, and so on. Hence it reaches the axon terminal, and it is then responsible for the release of the neurotransmitter. Now, what are the advantages of the saltatory conduction? This saltatory conduction only occurs in those neuron in those neuron where there are myelin sheath. In non-myelinated axons, there is no saltatory conduction. There is continuous conduction of the axon. Now the advantages of the saltatory conduction are that it increases the speed of impulse transmission because the most of the axon area that is you can say covered from the outside the environment, and the outside environment cannot actually affect the speed of the transmission. It also conserves energy, as depolarization only occurs in the nodes of Ranvier, as I've told you, because the ion channels, they are only present in these nodes of Ranvier. Now what happens that when the action potential it reaches the axon terminal, at the axon terminal it is responsible for the release of the neurotransmitters. When the neurotransmitter they are released into the synaptic cleft, they are going to interact with the receptors on the postsynaptic neuron, and hence this process of transfer message, it will continue unless a cellular response that is generated. Now how that happens, I'll focus on that in the next video. So if you like this video, please subscribe to my channel, hit the like button, and share it with your friends.